Welcome to our summer webinar series for 2023. I know it's been a little while since the COVID webinars that we were doing. I think our last big one was close to 2 years ago. And we've had a little bit of stuff on YouTube since then. Yeast Basics Part 2 with Nate Ferguson and some other stuff. But we thought, okay, it's been a little while. A lot has changed in the world of yeast. So it's time to provide some updates. We've been hard at work developing new yeast strains at Escarpment Labs. And developing and understanding better the yeast that we're using in brewing as well. All with the goal of controlling yeast flavor as well as controlling biotransformation, which I don't understand how it just keeps getting more and more popular, but it does. So biotransformation remains a hot topic, and we're going to break that down. What does that mean? And how do I use it in my beers? Okay, so this is one part of many part summer webinar series. So we've accumulated a lot of new information over the last few years here. So we've scheduled a few different summer webinars, so you can keep tuning into these. We're also going to be posting them on YouTube after the fact as well, so you can always check in and watch that after. Try to do a mix of some things that we're really excited about, like our house ale. So that's our new dry yeast. That's our first dry yeast strain. Fairly new to the market. The feedback's been awesome. So we have a lot of breweries using the house ale. The feedback we've been getting is that it's cleaner and faster than a lot of the other options out there. So if you're in a brewery and you're looking to turn around beer faster, especially in the busy summer months, it's a really great option to try out for your beers. Or even if you're just bored of the same old dry yeast that you've been using for years and years, it's a great option. So highly recommend that. We also have some less commercially viable content, such as our Brett Science update. So, regardless of how much it sells, we love Brettanomyces, and we're going to provide some updated information for you. We're also going to just give you some more information on yeast nutrition, non-alcoholic beer production, which has really blown up in the last year, as well as off flavors, as well as some of the other places that you can use brewing strains. And I thought I would provide an update. This is the most recent group picture I have of us. This is from the fall, so we need a new one. But we've grown a fair bit over the last 2 years. We've got a great team making great yeast across 2 facilities here in Guelph, Ontario, Canada. And we believe that fermentation should be easy. So that centers a lot of what we do. We're trying to make things easier for you, whether that is yeast cultures that just work the way that they're supposed to every single time, or whether that's knowledge around, how do I get the most out of my yeast? How do I get the most flavor and unlock all the potential of the yeast? And that's what brings us to today's topic. So I think you've probably asked yourself this question. I hope you have. How do you know what flavors to expect from a new yeast strain and how to use it in your brewery? But if you go and you look for answers to that question, sometimes the information is lacking. You might find some information on a homebrew forum or Pro Brewer or something like that, or Facebook. You might find some information from the yeast supplier, but oftentimes it's only the basic specs like attenuation, flocculation, maybe like a little flavor profile. There's not always a lot of information out there about individual yeast strains that tells us how do we actually use it? How do we use it to its best? So we realized there's a bit of a gap and made it one of our objectives this year to help address that gap and help empower all of you with information you can use to work with your yeast and make better beer, unlock flavors from it. And we do that through research. So we're not just guessing here. We do a lot of yeast research here at Escarpment Labs, and it's because we think it's legitimately the best way to help the brewing community. So we know that brewers want to know what flavors to expect from a new yeast strain, and the best way to address that is through research. We also know that you want to know how to optimize yeast flavor production using the different levers that you have to control that. So that might be temperature, that might be pitch rate, any number of variables. You want to know, like, if I ferment a little bit colder, what's the impact? If I pitch a little bit more yeast, like what's going to happen? Or, the glycol cooling on my tank failed. Is my beer going to be horrible? These are the questions that we're here to answer. And the answer is maybe. What tools do we use? So there's a nice combination of tools. So sensory analysis, human taste buds, human olfaction system, smell, taste, texture. is really, really important when we're understanding what yeast does, because at the end of the day, our goal is not a certain parts per million of acetaldehyde or diacetyl. Our goal is, does this taste good? Are people going to want to drink this? Are they going to want to drink more of this and enjoy the experience? So sensory analysis is really, really important. And we do a lot of that. It's a lot of beer tasting to understand if we change a variable, what happens with the yeast. But we also back that up with analytics tools. So through college and university partners, we have access to tools like GCMS, which is a great way. There's all sorts of different offshoots of GCMS. It's a great way to measure flavor compounds. If you want to measure esters and phenols and things like that, it's great for that and get a number. As well as HPLC. It's another great analytical tool to measure things like sugars or even some of the terpene compounds in a beer. And why do we do that? Well, and we also do genomics. Sorry, I'm getting ahead of myself here. We also do whole genome sequencing, and that's a really useful tool because we can analyze the whole genome sequence of a yeast, and that tells us a lot about what it might be capable of, because the genetics of the yeast are linked to its capabilities, to what we call the phenotype. So if we understand the genetics better, and especially as we continuously develop our understanding of how the yeast genes map onto the yeast flavors and the functions, we can start to predict and link, these traits with yeast, and it allows us to move a lot quicker. Because, for example, we can look for a very certain, gene mutation that we know might lead to a certain impact in flavor and function, without even having to, touch any flasks in the lab. So it's a really, really useful tool, and it helps us understand the yeast better. And we're often, when we're testing new strains and finding, interesting flavors, we're often able to go back to the genetics and ask, like, okay, great, we got this pineapple ester, but like, how is that working under the hood, and how could we reproduce this? So we combine all of these tools to the best of our abilities to develop our yeast understanding as well as our actual new yeast strains. And we're also just trying to understand, how does yeast make flavor? That's a big question we're trying to ask is, if we understand this better, we can control this, and in the future, you're able to get more tailored and more defined flavor profiles, and that's really exciting. And then also, we can understand off flavors better as well, and so understanding these things will help you make better beer, right? Ultimately, we all want to make better beer, better, faster, cheaper, tastier, basically, right? Probably not all those things, but some of those things. So just sort of center our discussion on beer yeast flavors, we have to first talk about yeast genetic diversity. So our brewing strains that we use in the beer industry are divided into different families. So there's two big families of brewing strains. One's called, this is easy, Beer 1, Beer 2. Beer 1 is most of the brewing strains that are used around the world. And these are separated into geographic families. So down below you have a British family, you have a Belgian family, and you have an American family. So there's actually, evidence you can see of these brewing strains evolving over the course of time in their geographic locations. And you can even see, for example, the American family is a bit of an offshoot of the British family, because there wasn't really, brewing yeast until British people came over to North America, for example. So you even see the history of human civilization and colonization and the genetics of the yeast. There's also a whole other family that we call Beer 2. That's up near the top here. That family is more closely related to wine strains. So they actually share some similar traits with wine fermentation yeasts. So they tend to be not quite as good at fermenting some of those complex sugars that are found in beer wort. But the trade-off is that they're really flavorful. They make a lot of flavor, and they've also found their own workarounds to handling beer malt sugars. So, for example, most of the diastatic strains, the strains that have that enzyme that can break down starches and dextrins into fermentable sugars, the Saison yeast, those are in this Beer 2 family, and those are also some of the most aromatic yeasts. So just to highlight that the genetics actually does tell us something about the flavor potential of the yeast. For example, the American strains tend to be more flavor neutral, whereas the Beer 2 strains tend to be some of the most flavor active strains. And by understanding the genetics of the yeast, we can understand why and how they make different flavors. And that's exciting to me because then we can control the flavors better. So what are some of the key flavors made by yeast? We start with alcohol. That's really where it all starts. That's part of the reason why humans have been fermenting with yeast for thousands and thousands of years is to make alcohol, ethanol, that thing that intoxicates us, gets us drunk, makes us social and less awkward. It's a really important molecule, and yeast helps us make it. But it's not just ethanol. However, ethanol is huge. Even beyond getting you drunk, it's really, really important to the flavor of beer. It's a flavor carrier. It helps to change and accentuate the flavor impact of a lot of the other different molecules in beer. Without alcohol, you have a totally different ballgame, and that's one of the challenges with non-alcoholic beer, for example. You don't have that flavor carrier. Just an example of other alcohols. One that's pleasant is called 1-octanol, or octanol. Actually lends a bit of a citrusy note if it's not in a beer in high abundance. You actually get it from some Kvike yeasts, and that can be nice. So not all alcohols are bad. Not all alcohols are like what we call fusel alcohols that smell like cheap vodka and give you a bad headache and a hangover. Some of them can be beneficial. Another example is glycerol. Glycerol is flavorless, but it modifies the body and the mouthfeel of the beer. So that's another important alcohol made by yeast. And that's just made from sugar, right? Yeast will convert sugar into alcohols. In the case of our beer ferments, it's usually like mostly ethyl alcohol, ethanol, but we do get other stuff, and those are themselves flavors, and they are also precursors of other flavors. And that's what brings us to esters. So esters are one of our sort of categories of interest here when we're talking about beer yeast flavors. And that's because these are really lovely, fruity, floral molecules. They can taste like banana or pineapple or roses. There's quite a lot of interesting esters out there. We typically divide esters into fatty acid esters like ethyl hexanoate, which is our pineapple. I love that molecule personally. And acetate esters like isoamyl acetate, banana. I'm personally not so much a fan of the banana, but I know some people are. And I will show you how to get more of it in your beer if you so desire. Esters are made. You got to start with an alcohol, and basically the combination of an alcohol and an acid. So my high school biology teacher had this drilled into my head that Esther, the party girl, combined acid and alcohol. That's how you know. That's how you learn how it's made. Literally, I learned that like 15 years ago, and it's still in my head. So shout out to my high school biology teacher. But yeah, really, you have an acid on one end, you have an alcohol on the other. So it could be, for example, a fatty acid like caprylic acid. And then ethanol, those things combined together, you get ethyl hexanoate, and it tastes like pineapple instead of like goats. So really, really cool stuff that yeast helps do. On the opposite end from esters, we have phenols. So phenols are basically coming from, in the case of beer anyway, they're coming from grain. So grain contains these phenolic acids, which in the plant itself, in your barley, wheat, rye, those are stress protectants. They actually help basically protect against pests and other microorganisms like molds and things like that, because they're actually toxic to a lot of these organisms. So that's true for yeast even as well. So a yeast that has the ability to get rid of this stuff will. And what they will do is they'll take that toxic phenolic acid and they will convert it into a flavor active molecule. So one example is 4-vinyl guaiacol. That's our classic like clovey kind of character that you get in a Belgian strain or a Hefeweizen strain. In a lot of beers, this is unwanted or unpleasant. So you often hear this called POF or POF, phenolic off flavor. I think, yeah, sometimes, walking around Escarpment Labs, we just call it POF, but it's useful to have the context. And it's not always an off flavor, right? Like it is an expected flavor in a lot of Belgian style beers, as well as German wheat beers, and even some other traditional beers like English stock ales and things like that. But it's just coming from phenolic acids in your malt and grain. So like that itself is kind of an example of biotransformation. We think of biotransformation as being a hop thing, but there's also biotransformation of precursors in your malts as well, and phenols are a great example there. We also have acids. So yeast can actually produce a few different types of acids. That includes succinic acid and acetic acid. Usually those are at pretty low ranges. Those can impact the overall taste profile of a beer. Some yeast strains do produce more acids than others as well. So, for example, Kvike strains, Saison strains tend to be on the high end, and then lagers, English ales tend to be on the low end of that. There are specialized acid-producing yeasts that can make lactic acid as well. So Lachantia, for example, our lactic magic is an example, and there's also some bioengineered strains out there as well. And why this is important is that acidity produced by the yeast can impact the taste perception because of pH differences. And sometimes that is something that needs to be dialed in. Like, it's something we've run into a lot with Kvike, where it's helpful to adjust the actual wort production and water profile so that beer isn't too acidic. So if you're making like a crispy lager, it's not, coming across as too tart and thin. And there's lots of ways that you can adjust that. Okay, so that leads us to biotransformation. So biotransformation, the way that we're going to define it, it's not just dry hopping in the middle of the ferment. No. We define it as the process where yeast change less desirable or flavorless compounds into more desirable and bold flavors and aromas. That's really what you're looking for when you're talking about biotransformation, right? You're looking to get something from nothing or something from something less and add a little bit of oomph, a little bit of flavor from your yeast. So in other words, you're bringing your other ingredients to life by yeast. The cool thing there is that, yeast will do that for you as long as you treat it right. And so it's a really, really beautiful combination. If you sort of understand some of the basic principles of biotransformation, which we're going to get into. So I'm going to talk about one, terpene biotransformation. This one does not get quite as much love right now compared to thiols. I know thiols are the hot thing right now, and we're definitely going to get into thiols as well because they are pretty cool. But terpenes are just as cool. So, I'm going to be arguing the case for terpene biotransformation and playing around with that in addition to thiols. So terpenes are compounds that are often found naturally in your hops. That's your primary source of terpenes to be biotransformed is hops. So hops have all sorts of different terpene compounds that have different flavors. So it might taste like pine, it might taste like roses, it might taste like lemon, or even like cannabis. These are all terpene compounds that are found naturally in the hops. And different hops have different terpene profiles, and it's a huge part of the flavor profile of an individual hop. Especially when you get into pretty much any like new hop variety has a pretty specific terpene profile. It might have some other esters and thiols and things like that that are giving it a little bit more of a specific character, but it's a huge part of the profile. So it's impossible to ignore. And what's really cool here is that yeast can both unlock and it can also transform terpene compounds from your hops. So how does it do that? What it can do, for example, is it can use an enzyme that can release bound terpenes from the hops. So you can have terpenes that are both bound and unbound, and this is going to be a similar story with the thiols. There are yeast enzymes that can break these down. They're called beta-glucosidases or glycosidases. Some yeast strains are a lot better at this than other. In our experience, most Saccharomyces yeasts are not that good at that kind of terpene release. When you get into Brettanomyces, they tend to be a lot better at that. There's some really good research on that out of Belgium. And then there's also the conversion of free terpenes, which you're going to find in abundance in your hops anyway, to other compounds by yeast. So yeast can take one terpene and it can turn it into other ones. And depending on the terpene you're starting with, and also depending on the yeast strain, you might get a different terpene profile. So this is one of the key things that differentiates different types of yeasts is the type of terpene biotransformation they can do, as well as the amount of terpene biotransformation they can do. So for example, you can start with something like geraniol, which to me really just does taste like rose water kind of thing. It's not that pleasant on its own. It's also found in abundance in pretty affordable hops, like Centennial is a great example, pretty high geraniol. But yeast, especially with the right yeast strain, can convert that into A bunch of others types of terpenes. So citronellol is a great example. It tastes more like citrus, lemongrass, lemon lime kind of character. And what's really cool about citronellol is it's not naturally found in hops. So if it's present in a beer, it's the result typically of biotransformation by yeast. So we end up using that as a marker for biotransformation. And it's also a great example of like, okay, you don't actually get this specific citrus in hops, but when you add yeast, you actually can. You can turn rose into something a lot more bright and citrusy through yeast selection. There's also some other nice molecules like nerol, which also has that kind of lemon lime character, and linalool, which I actually really like linalool. If you smell mosaic hops, they're typically quite high in linalool. On its own, it kind of smells like Fruit Loop cereal because I just assume that Fruit Loop cereal are just flavored with linalool, but that's a great sensory standard if you're interested in linalool. Same thing, lavender has a lot of it in it as well. So some people might perceive linalool as being more lavendery. And I would recommend trying different yeast for this. We've got some data in this presentation, but I would certainly look at Hydra. That's a strain that was developed specifically for juicy IPAs. And a side benefit of that strain is that we selected two parents to hybridize to make Hydra. Both of those parents are quite good at the terpene biotransformation. Hydra has maintained that trait, so it's quite good at the terpene biotransformation. So you can put in hops that are quite high in geraniol, like Centennial. Another great one is Strata, where Strata can almost be too dank on its own. With the right yeast, you get the biotransformation, you get really, really nice citrus, really changes the profile. So that's something I recommend. There's a big storm happening outside, by the way. So just as a heads up, there might be some background noise. And then we get into thiols. That's kind of the hot thing right now. And it's because they're really, really interesting molecules. And there's a lot of development in the yeast world for these. So certain yeasts are capable of unlocking thiol aromas from flavorless precursors in both your malt and your hops. And what that can do at the end of the day is it can make a beer taste more tropical. So just like with the terpenes, we can have bound thiols. They're basically bound up in a form that's not flavor active. In the case of the thiols, usually you have a thiol molecule that's connected to an amino acid or a polypeptide. And when it's connected to that amino acid, the yeast can use an enzyme. It can basically break that apart. Yeast says, great, I get my cysteine. I can go and make proteins. And we say, yum, this beer tastes like grapefruit and guava now. So it's really, really a nice synergy for both of us if we do it right. That is dependent on the yeast IRC7 enzyme in our normal sort of brewing strains. And part of the challenge here with thiol biotransformation is that most brewing strains don't have an active version of this enzyme, or they have a poorly active version of this enzyme. So you do have to develop new strains typically to unlock thiol biotransformation. There are different approaches to doing this. You can overexpress the gene in a yeast, or you can, there are some examples in the US where they've put in an enzyme from bacteria into the yeast, and you get like even more thiol conversion. And then there's also leveraging natural strains that are quite good at thiol biotransformation as well. So that's an example of what we did to make Thiol Libre, which there's a new version of Thiol Libre out now. Highly recommend it. It makes super delicious beers. I've actually got one here right now. One of the things I really like about Thiol Libre is that it's quite good at making the 3-MHA thiol, which requires both this release of bound thiols and also ester production. So the background of Thiol Libre is a wild yeast crossed with a Kveik, had pretty naturally good ester production. So you end up being able to produce pretty nice levels of that passion fruit guava aroma from those flavorless precursors. So just for example, the beer that I'm drinking was made with Cascade in the mash, but it tastes a lot more tropical than it should. So highly recommend trying that out. And that's basically how this thiol release works in most brewing strains that have been developed for this application. So just digging into a little bit more of these individual flavor compounds. So we have some different key alcohols. Like we talked about how yeast makes alcohol that intoxicates us, but they also make alcohols that have flavor. So isoamyl alcohol and phenethyl alcohol are two examples of that. They both actually have some positive flavor traits in terms of complexity. If they're not at extreme levels, they can come across either off-putting or boozy if they're at really high levels. Yeast also makes all sorts of different esters, so I just wanted to highlight a few of them. The isoamyl acetate, that's the banana ester. You love it or hate it, basically. We have the phenethyl acetate, which is sort of more of a floral honey kind of. To me, it kind of just smells like yeast, but it's sort of a characteristic aroma of yeast, and that's why. As well as ethyl acetate, which is quite nice. It can add sort of a wine-like complexity to a beer if it's there in low quantities. It can come across as solventy if it's there in really high quantities. We also have what we call the fatty acid esters. So these are ones where we've taken an acid and alcohol, and in this case, it's a fatty acid, short chain fatty acids, medium chain. Ethyl hexanoate, this is one of my absolute favorites. I really like when it's in a beer in abundance because to me, it smells like pineapple and tropical fruit. Really, really nice flavor from that one. It's nice to have it mixed in with other stuff, but it's a really nice one to make beer taste good. Ethyl octanoate, not far off, maybe a little bit more stone fruity kind of character, and ethyl decanoate is another similar molecule that comes across more like orchard fruit like. It can add a lot of complexity, especially when you get into like some of the darker beers. I find like dark malt, like a stout, with a yeast that produces high ethyl decanoate can be a really, really nice combination. And then citronellol, what we talked about, that's the terpene that we basically need yeast to make. Really, really nice one that has citrus lemongrass character. And then, of course, our phenol as well. So the primary phenol that we look at in beer is 4-vinyl guaiacol. That's the clove and black pepper that's made in wheat beers and Belgians. There are other phenols that Brettanomyces will make. We won't get into those yet. Okay, so just to highlight, all this information is available both in print format if you're a customer or visiting us at a conference, as well as in a PDF. That's our yeast flavor and biotransformation guide. Make sure to check that out. I'll have a link at the end as well. But just to highlight, if you don't have to take super detailed notes because you can find this information on our website if you need it. Okay, so what does yeast aroma data look like? This is an example of basically taking a whole bunch of our strains and throwing them in a cage match together to figure out which one tastes the best. What produces the most of each flavor compound. That's basically what we did. We actually did this study about 4 years ago now, but we've been working on analyzing and sharing the data ever since, basically. So really what we're seeing here is the spectrum of yeast flavor potential across quite a few of our brewing strains. So we were able to include some of the newer ones in there as well. And you can see, the diversity both in terms of intensity of flavor and the flavor profile. So we'll get into some of the specifics, but just as an example, you can select yeast for high intensity character, or you can also be selecting yeast for neutral flavor profiles and letting the other ingredients shine through. There's no better way. It really just depends on your objectives, the type of beer that you're making, all these things, right? You probably don't want to pick a yeast with like crazy banana ester if you're making a Czech Pilsner, right? And conversely, like if you're making a Saison, you probably do want to pick a yeast that's got a lot of flavor impact. So one example, if you're really into banana, what yeast do you pick? So here's the data on isoamyl acetate. So you can see some of the strains just don't even produce a detectable amount of that aroma compound. They're way below the sensory threshold. In all of these graphs, we've scaled the actual concentration of the compound to the sensory threshold to just better approximate, what is the actual impact of this. And if you want something super banana-y, you have some options, right? So you really see the Saison yeasts punching above their weight. Some of the new strain, like Jotun is another example. And also some of the Kveiks, you can see Hornindal does produce some of that banana ester. So if you're looking to push that, understand that it's not just Hefeweizen yeast. You can get that flavor from a pretty diverse range of yeast, whether it's Saison, Kveik, just by choosing the right strain. Something to also highlight is that you can control this flavor, regardless of what yeast strain you choose. A wort that has a higher glucose content will have a higher isoamyl acetate content. So, for example, if you're making a Hefeweizen and you want to get more of that banana flavor, consider adding a little bit of glucose. I mean, obviously, that's not going to fly in Reinheitsgebot, Germany. That would be verboten. But anywhere else, you could just add some dextrose. Otherwise, you can play around with your mashing and make a more glucose-rich wort, for example. And typically, having more glucose will give you more of that isoamyl acetate banana ester. So that's one way to play around with it. If you've got a Hefeweizen that's too clovey, try adjusting the amount of glucose in the wort, and you might enjoy the results and get a little bit more of that banana. We have ethyl hexanoate. So I talked about this earlier. This is one of my favorites, that pineapple ester. The beer that I've got here has a decent amount, which I'm very excited about. And there's quite a range for this one as well, from basically nothing to several times the sensory threshold. So when you look at the data, you see, in this case, our Denzale really punches above its weight. We know that from experience. That's quite a nice sort of pineapple-y strain. It does have the phenol production as well. But if you're looking for a fruity Belgian strain, that's one of the best options. Again, our Hornindal Kvike really punching above its weight. It's kind of an ester powerhouse. And a lot of other options. And you actually see some surprising ones, like Autobahn Lager is pretty high there as well. So that's an interesting strain, potentially. And then if you're not looking for that pineapple, you have options as well, like the Weizen 1. We love that strain because it's, if you do make a glucose-rich Hefeweizen wort, it's pretty focused on that banana clove, and not a whole lot else going on, which is great if you're shooting for that sort of competition, style, classic Hefeweizen. Something to highlight here for those fatty acid esters like ethyl hexanoate. Pushing the temperature higher can push more of that ester. So we got some data later as well, but typically you can get more of those esters if you push the temperature higher. That's not always true for every ester, as we'll see, which is kind of interesting. And our terpene biotransformations, we got one more green graph. Beta-citronella, once again, that is the one that has to be made by yeast. It's not typically present in wort extracted from the hops. So it's a really good way for us to measure terpene biotransformation by the yeast. And once again, we see a pretty wide range, right? From several times over the sensory threshold down to basically nothing. So you have a lot of options to play with. If you want to preserve that kind of more floral, aromatic, and terpene profile of the hops, then you can choose a strain with lower beta-citronella biotransformation. Or if you really want to push that citrus, you can pick a strain with higher terpene biotransformation. So you can see Hydras in there near the top, some of the Saison and Hefeweizen strains. Überweizen, compared to Weizen 1 is a fair bit more intense. So that strain, you will get more citrus compared to the Weizen 1. That's one of the key differences. And then again, Hornindal, just punching above its weight, absolute flavor powerhouse. So, shout out to the Hornindal Kvike. It's always doing some cool stuff. And then some of the other strains that are commonly used for IPAs, like Vermont, Cerberus, they're up there as well. Some of the other strains that are also super common for IPAs, like Foggy London, for example, and Cali Ale, are not so impressive with the terpene biotransformation. So, moving forward, there's probably some room to improve with some of those strains and some of those profiles in terms of the terpene biotransformation. If we want to push more flavor, with a beer that's made using something like Foggy London, we probably have to make a new yeast at the end of the day. We can also look at these profiles by category. So, one way to think about this that I think is really useful is the yeast phenolic or non-phenolic, because that does have a pretty big impact on the flavor profile. Yeast with noticeable phenols is, I think, a lot of the time going to be perceived by most people as kind of Belgian-y or Hefeweizen-y. So it does have an impact, and it can also mask some of the fruity flavor compounds as well. So it's useful to us if we're looking at designing a recipe. Are you looking for a phenolic yeast or a non-phenolic yeast to start, and then sort of digging into the individual flavor profiles, right? So if we're making a Belgian triple, we probably don't want to go for any of these strains. But if we're making an IPA or like looking for a new house yeast, then this is probably the strains that we would want to be looking at. So if I'm making an IPA and I just want to absolutely maximize yeast flavor production, I might go and look at Hydra or Cerberus or Hornindal Kvike. If I'm looking for something more neutral, I might play around with Cali or English Ale 1, for example. Kvikes here are a little bit misleading because in this case the temperature is like 20 Celsius, and as we know, Kvikes are a lot fruitier and are often fermented a lot hotter. So you will get quite a lot of esters from Voss and Laerdal, for example, if you run them at hotter temperatures. But you actually can make super neutral beers with some of those Kvikes at lower temperatures. That's the whole story behind our crispy, for example, is developing a Kvike that can be pitched at 20 to 25 Celsius and make a lagery beer really fast. So lots of things to play around with, and you can kind of, okay, what do I like, what do I not like in terms of these aroma profiles, and pick a yeast based on it. With our phenolic yeast, so say, for example, we're designing a Belgian beer, because these are the yeasts that are great for those Belgian beers. We also have some options to play with. In general, what's kind of interesting is that the phenolic yeasts are more aromatic than the non-phenolic yeasts. For whatever reason, a lot of it is because a lot of these are in that beer 2 family that's more closely related to the wine yeast. It's just a different family of yeast genetically, a different background. But you do get a lot of real aroma powerhouses here, and a lot of good platforms with these phenolic yeasts to enhance other ingredients. So if you think of like a wit beer, for example, I didn't mention this earlier, but wit beer, the coriander and orange peel, that will also contain precursors that the yeast can biotransform, and you might get more aroma out by choosing a certain strain. So if you look, for example, Uberweizen and Farmstand Saison here, they have quite a lot of beta-citronellol production in pink. So you might be able to push more of that citrus from your coriander, for example, in a beer that includes that. You can also look at the ethyl hexanoate, right? Look at Ardennes, and if you want something a little bit more pineapple fruity, to sort of downplay some of those phenols, it can work really, really well. So if you're looking for like a fruitier Belgian beer, that would be a great option. So you have a lot of different flavor profiles to work with. The lagers, kind of the opposite story. Generally, lager yeasts are less flavor active than ale yeast. That's kind of their whole MO. That's lager. They don't taste like much. And the other thing is that lager yeast itself is the result of really just one or possibly two hybridization events somewhere in Central Europe around 500 years ago. So for that reason, the diversity, because there's this like evolutionary bottleneck, the diversity of lager yeast is pretty low. So the flavor diversity is also pretty subtle in general. So lager yeasts are a great platform for other ingredients to shine, but they're typically not the greatest platform for their own flavor production and biotransformation. So if you're really looking to push like the essence of your hops, the essence of your malts, lager strains are a great option for that, but not necessarily the best option for like enhancing any of those traits. So there's also a lot of room for improvement there too. It would be really cool to see some lager strains that can potentially, still ferment cold, but push more aroma and more flavor in the beer. So yeah, with some of that knowledge, and again, that's quite a bit of a knowledge dump there, but you can find this in a PDF or physically if you come and track us down at a conference. With this knowledge, you can select the right yeast for your desired profile. So that's really what we're trying to help you with, is give you as much information as possible so that you can select the right yeast, predict the outcomes in your fermentations, and really get to your target flavor profile faster without as much trial and error. So just getting into some of the things that I mentioned earlier, some of the fun stories. So you actually can control your ester production by yeast with your temperature. So I kind of alluded to this earlier about how some esters are produced more at high temps and some are not. And that seems to be true for the acetate esters. You actually, hotter isn't necessarily better for these. So if you're looking at like the banana, for example, the grape, those kinds of aromas, going to a hotter temperature might actually be counterintuitive. So, we don't have a massive selection of strains to look at here, although there is more data coming. We do see in general for a lot of these strains, the maximum is, not at the high end of the range that we looked at. So this is also, admittedly an experiment with Kvike where we were going up to like 40 Celsius, which I think is over 100 Fahrenheit. So pretty extreme. But even so, if you look at a lot of these strains, especially the strains that might be used more commonly for this type of ester production, like St. Lucifer, for example, you actually see more of this ester at lower temperatures. So that's kind of interesting. If you want to maximize the banana, with a Belgian strain or with a Hefeweizen strain, I would actually experiment with reducing your temperature and try it out and see how that works for you. You might have some good results. I've heard some great results with Hefeweizen strains fermented around 16 Celsius, for example. It's a bit of a different story with the fatty acid esters. So that's our pineapple, orchard fruit kind of profiles. It is also kind of strain dependent. You can even see here the magnitude of production between some strains, like Cali just doesn't make very much. You compare that to some of the Kvikes. The Kvikes are absolute powerhouses in making fatty acid esters. So that's one of their strengths. But you see for some of the strains, it just keeps going up with the temperature. Like Voss, you get the most at around 40. And what's kind of cool there is the guy that it comes from, Sigmund, he pitches at 42. These farmers know what they're doing. Hornindal, same thing. Maximum at 30. Guess what temperature they use on the farm? 30 Celsius. So don't discount the farmhouse wisdom with any of this Kvike stuff. It's often very, very real. And that's often a way to understand the yeast better and get better flavor. So those are some pro tips. If you're looking to maximize those esters, you might be able to play around with the temperature. Of course, individual esters you might see maximize the different temperatures. So if you look at this data, in a lot of cases, you actually see that ethyl hexanolate, our pineapple, is often in higher abundance at the lower temperatures. So once again, you might have better results. I think the common wisdom is that you have to, if you go hotter, you get more esters. That's not necessarily true. And certainly the temperature changes the balance of different esters that you find in your beer. And if you're looking to push the pineapple, once again, colder might actually be better in some cases. That is strain dependent. What does pitch rate do? So this is another important one that you have control over as a brewer. You can use higher or lower pitch rates to get more or less flavors from your beer. So we did test this. We tested this a few years ago, both with Kvike strains and also with a Vermont ale strain. And the results were, in essence, mixed. So you can see this is a PCA plot, which is like a map of the flavors. If you look on the right-hand side, you can see, what different traits are creating that map on the left. So near the top are Vermont at our low pitch rate. That would be associated with beta-citronellol. So in some cases, you do see some differences in pitch rate in terms of our flavor and biotransformation, although it's very strain dependent and it's not very predictable from strain to strain. So there's probably still some work to be done there. I typically don't recommend pitching Vermont at 1/10 of the normal pitch rate because you're also going to get isobutanol, which is a fusel alcohol. Probably not a great idea. But with the Kvikes, certainly if you look at those ones like Hornindal and Voss, going to the right, you see basically more esters with those. So that is a way to get more flavor out of the Kvikes. You can play around with underpitching Kvike yeast and get a different flavor profile from it. Certainly it's not the same, right? You do see differences here. And then Brett. I threw this in because we're going to be talking about Brett in the next webinar. But I just also want to highlight that despite all the stuff that's going on with new developments in Saccharomyces yeasts and new hybrid strains and bioengineered strains, Brett remains an absolute flavor powerhouse. It's a flavor bomb. Brett can produce a lot of these flavor compounds in greater abundance than Saccharomyces can. That being said, there are some trade-offs, right? Brett on its own in primary fermentation doesn't ferment very well. It's very unreliable. This analytical data is from Brett primary ferments, so those would have taken, between 21 and 28 days to finish fermentation, whereas, a normal Saccharomyces should be done in about 7 days. So it is quite a lot slower, but you do get a lot of flavor. The trade-off with Brett is that the slow fermentation, and also it does produce those phenolics. So that sort of smoky medicinal, it produces a wider range of phenolics than normal yeast does. So it's not just the clove, you get more of a smoky thing, or even that like medicinal or, Band-Aid kind of thing. And acknowledging that those can also mask some of the fruitiness. So with Brett, it's kind of a bit of a balancing act there. So, some work to control the phenolics with Brett can also pay off in terms of promoting the aroma compounds. So that's always something to play around with or consider as well. But yeah, like for esters, even for the terpene biotransformation, you get a lot out from Brett, and so it's certainly worth playing around with it. Even though things have moved towards more efficient yeasts over the last couple of years. So what are we going to brew next? I hope you feel inspired from some of this information. And if you do have any questions, you can always ask us. I just wanted to highlight once again our Thiol Libre, because we did recently release an updated version of it. So it's a yeast that we produced through yeast hybridization, yeast breeding, to be better than typical brewing strains at Thiol biotransformation using the IRC7 enzyme. So this strain does have a background of Kvike and wild yeast that makes it have a really wide temperature range, really easy to ferment with. The beer that I'm drinking right now has been produced grain to glass in 8 days. It was done fermenting in 4 days. I gave it a bit of time to clean up. Cascade mash hop and some malt from Shoreline Malting out in Prince Edward Island. It's a pretty aromatic beer. It has that sort of passion fruit thing going on. It has a little bit of pineapple. I thought it was aromatic enough that I didn't even need to dry hop it. So yeah, again, grain to glass in 8 days. Possibly the easiest beer I've made in years, and I think it tastes pretty good, and it's going to disappear pretty fast. So just want to highlight some of the things that Thiol Libre can do. It's not just like super crazy IPAs. You can even make a pretty, like, this is like almost like a blonde ale, and really, really tasty. But yeah, some of the benefits of Thiol Libre. You can get more out of your boring hops like Cascade. That's true of any yeast that's developed for enhancing thiols. There's a lot of cool stuff they can do beyond just like the hazy IPA world. Of course, they have applications there too. You can make your sort of light summer beers, or you can make a big DIPA, and get more aroma out. One of the nice things about Thiol Libre is that in addition to the thiols, it's also quite a good producer of ethyl hexanoate, the pineapple ester. And those things I find are pretty synergistic. The tropical character of the thiol, like 3-MHA, alongside the tropical characteristic of the yeast ester. They're very synergistic. They work together, and you get a really nice flavor impact. And this version of Thiol Libre, the previous version had some potential to produce phenolics, depending on the base malt. That's no longer an issue. So we've been able to address that and make it easier to use. Okay, so we're just wrapping up. If you want to learn more, we have lots of resources. So you can check out our blog. There's more than 100 posts on there now that can help you. We have our knowledge base. So that was launched about April of last year. So that's where we keep a lot of specific chunks of information, things that are not necessarily big enough for a blog post, but that are still important for us to share and make accessible to you. So if you want to learn more about pitch rates, or how much free amino nitrogen does my yeast need, how do I troubleshoot fusel alcohols, that's all on there. Check it out. And then, of course, our YouTube channel. So in addition to this webinar, there's tons of content on there, tons of useful stuff that's going to help you be a better brewer, make better beer, and definitely check that out as well, especially if you're a visual learner. I know I tend to just like learn from YouTube. And yeah, also highlight, we got a bunch of summer webinars coming up. Check those out. Make sure you sign up on Crowdcast. I've got a link there. Sign up ahead of time, and I will also promise that this one won't go off without any hiccups. I think they changed format or some kind of bug happened, but I'm glad we were able to get this one going, even though there was some small delays. And thank you for your patience as well. And yep, once again, you can find our yeast flavor and biotransformation guide on our website. You can scan that QR code, or you can just go to our website and find it and download it. That gives you all the information I showed you here and more about yeast flavors, biotransformation, everything you want to know, basically. And that's it for me. If you have any questions, I'm going to stick around for a little bit. If you have any questions, feel free to ask them either in the chat, or there is also like a dedicated question system in Crowdcast on the right-hand side as well. So you should be able to go in and type in some questions, and I'll be happy to answer them for a few minutes. Thank you so much. Question from Matt. Evergarden really doesn't produce any isoamyl acetate. It probably does depend on the conditions. I've certainly tasted a bit of banana off of it before. There is also isobutyl acetate, which is, well, you take acetic acid and butanol, or isobutanol, and combine those together. Very, very similar to isoamyl acetate, very similar aroma profile too. It's also kind of banana-y. So we didn't measure isobutyl acetate in this experiment. So it's possible that if you taste some banana with Evergarden, that maybe it's making that ester as well. So that is a limitation with some of these studies. You know, we're not necessarily measuring absolutely every flavor compound under the sun, although I would love to. So there's always room to sort of go back and refresh these things too. One from Colin. Is there a dose correlated response of a glucose, assuming dextrose addition, to increase isoamyl acetate? There is like an upper limit, I will say. I think that there is, I don't recall exactly. The presentation itself was from data presented by Lalmond about 6 years ago or so. So I would look for that one. There might be some scientific data as well. But yeah, there's certainly a certain, like, it's not just going to keep going up with more glucose. Like, there will be sort of a point of diminishing returns, right? So I believe it was recommended, like, if you can sub out 15-20% of the fermentable sugars with dextrose, you can get an appreciably higher concentration of the isoamyl acetate. Brandon asks, which one is bubblegum? Is that high concentrations of isoamyl acetate, or is it another compound? It can be a combination of things. So when I was talking about Evergarden and banana, again, some of those isobutyl esters, isobutyl acetate, can, like, to me, those come across as that kind of bubblegummy thing more so than banana. So that's something that wasn't really measured here. Often I find that combination of some of those esters with like phenethyl acetate as well, that kind of floral yeasty aroma gives that like bubblegum perception. Generally, it's some kind of acetate ester, more so than fatty acid ester when we're talking bubblegum. That's characteristic of a lot of Belgian strains and wild strains and things like that. Alrighty, I don't see any more questions, so I think we're going to sign off, and I will hopefully see you on August 8th for our talk about Anamyces.